course, to the computer. Can just one of you guys let me know? Can you see my screen?
Yes.
Thank you. All right, guys. So today we're going to talk about hepatitis viruses. These are a weird, let's say, kind of interesting group of viruses. First of all, hepatitis viruses, they're not a family. They're just technically a group of viruses that we put together purely because they're able to go all or have a specific tropism towards our hepatocytes, hence our liver. And then, of course, they induce the inflammation of it, hence hepatitis. And we can put them in five, let's say, kind of viruses: hepatitis A, hepatitis B, C, D, and E. And the thing about them is that the only common thing between them, the only common thing between these viruses is they are structurally very, basically, and genetically very different. They have different families and different, some of them are RNA, some of them are basically DNA viruses. The only thing that is actually making them, let's say, part of the hepatitis viruses is that they have a hepatropic, basically kind of, let's say, tropism. They will only go to the liver cells because their host cells or the receptors they bind to is specifically in the liver. And something that is interesting about these cells or about these viruses is that they do not cause cell death. They are actually quite harmless in that kind of sense. When they get into basically your cell, most of what they do is kind of stop a bit the kind of cell's ability to translate into proteins. But it's not that actually kills us. It's our immune system. All of the symptoms and all of the complications that we get from hepatitis viruses is purely from your immune system, and specifically cytotoxic T cells. So cytotoxic T cells, they become, of course, let's say, white. They basically get taught to go kill our hepatitis virus. So they love to go to basically infected liver cells that have the hepatitis viruses, and they keep bombarding it. They keep hitting it, and usually, if the hepatitis virus is stubborn and stays for prolonged periods of time, our immune system is going to keep bombarding it, and it's going to cause a lot of complications. And of course, the most thing that we know is cirrhosis, the scarring of your liver. And then this could actually turn into hepatocellular carcinoma, which is basically liver cancer. So, overview of the hepatitis viruses, as we said, they're very different. They have different RNA or DNA. So, for example, the only difference mostly is coming from hepatitis B. Hepatitis B is a double-stranded DNA, while the others are all single-stranded RNA. Some have an envelope and some don't, actually. Their mode of transmission and their ability to cause chronic infection. So, hepatitis A and E, these guys are quite well-known hepatitis viruses. They usually go through the fecal oral transmission. And these guys are quite mild. They do not cause, per se, they don't do any chronic disease. They're quite easily removed and quite easily, actually, you get a good immune system against them. But the other ones, B, C, and D, these are, you can basically get them from blood, sexual, or prenatal transmission, technically. So this has to do with more kind of bodily fluids. And these ones can cause actually chronic inflammation. So those can be a bit more, let's say, dangerous. So let's go through all of them. Let's go start with hepatitis A viruses. So hepatitis A viruses are non-enveloped viruses that have a single-stranded positive sense RNA. They basically are non-enveloped, so that makes them extremely good and stable in the environment. So in generally, they can actually withstand very harsh conditions. And one of them is the acidic pH in our gastrointestinal tract. So basically, usually when we're actually infected with basically with hepatitis A virus, we actually contract it through our mouth. This could be through contaminated water, or it could be contaminated basically food. Most of this food actually comes from oysters. Usually, Yeah, for example, I'm a lover of oysters. I really love oysters. When I was living in the US, they used to have these kind of all-you-can-eat oysters kind of deals. But I used to be extremely afraid. Like I used to go to really good restaurants, pay that extra amount of money because a lot of oysters, if not well taken care of, they can contract hepatitis A virus, and hepatitis A virus is not something that you really want to be infected with. Even though it's in some sort of way, let's you only get it once and you get cured quite easily, the symptoms are quite annoying. So the thing about hepatitis A virus, it only has one serotype. So this is a very good advantage for us because one infection, just get infected once with it, you get a long-lasting immunity. So in per se, usually you only get infected once in your lifetime. Let's see, just quickly something on the chat. Why are kids at high risk after birth? That's a very good question, William. Actually, in general, it's mostly so it depends, of course, where they're born, and of course, because there's certain endemic areas where the water is very highly contaminated, because usually it goes to the fecal oral kind of routes. So hepatitis A is not really, let's say, that it's not really given prenatally. So that's one. So two is mostly the endemic areas that they are. But in generally, kids, especially babies that are just born, do not have a proper, let's say, T cell response or B cell response, and they usually can die from just a simple hepatitis A virus because they have a premature immune system. So the transmission, as I said, it usually happens to the fecal oral route. So it's contaminated food or water. And the thing that makes hepatitis A virus extremely, let's say, successful at infection is that once you contract it, basically the hepatitis A virus, it can stay there for 2 weeks and keep shedding basically through basically our, let's say, waste for 2 weeks without us getting a single symptom. So we can be asymptomatic for 2 weeks, but we keep shedding so much of the virus that can go into the sewer waste or basically in some sort of way. Uh, yeah, well, it's mostly to the sewer waste that it usually goes through. And of course, this can be a huge issue in basically areas like, the camps, institutional schools, where you have basically a water source. And of course, it really will just spread. Course contamination through food, specifically, like I said, shellfish like oysters. They basically usually these ones, why are they the ones that actually have it? Because they actually can be living within the polluted waters that, the hepatitis A virus is found. So again, usually you get it through drinking the water or eating basically something that's contaminated with hepatitis A. And as I said, it's because of it doesn't have an envelope, it's able to basically go through our stomach and our colon and withstand all these different types of basically pHs, like in the acidity in our stomach. And once there, it basically goes through the blood into your basically liver. So once in the liver, and of course, it doesn't do the damage. It just creates an inflammation that brings the cytotoxic T cells. And the cytotoxic T cells are there, just keeps bombarding your liver cells that causes hepatitis. So the good thing about HIV, basically, sorry, HIV, HAV, so hepatitis A virus, it does not establish chronic infection. It's very easily removed. Usually, most basically, your body is able to clear it out, and you get a very strong immune response that usually you do not get infected again. So what happens technically, well, if you look at our serology, so if you look at more at our antibodies in our system, usually what we see at the beginning is that specifically right when we have symptoms, basically we have basically the hepatitis A in our stool, the symptoms, and especially jaundice. Jaundice is basically this yellow discoloration, and this usually happens when our liver gets damaged. It releases basically bilirubin. Bilirubin is basically a protein that sticks to basically our skin or basically to our basically kind of eyes that gives us this yellow color, and this is very indicative of liver damage. So when we get these symptoms, we have basically a lot of IgM antibodies. So remember, guys, IgM is an antibody that is usually only secreted at the beginning of an infection. So we use a lot of IgM, tell us a lot, is this acute, this infection or not? Because then after a time, it goes down. And as it goes down, we have IgG antibodies. And these IgG antibodies that are produced are lifelong. So this is what's going to protect you for any future infection. So clinical features of hepatitis A, you have fever, loss of appetite, nausea and vomiting, jaundice, which is this yellow skinning of the eyes, and you have this dark urine and pale stools because technically your liver is messed up in general. So again, this usually resolves around 2 to 4 weeks. So it is a stubborn virus. It does stay quite some time, but again, it's usually self-containing, and basically you get a lifelong immunity after it. So when it comes to diagnosing it, it's quite simple. It's usually most of the time you look at IgM antibodies. If you have IgM, you know that this person has an acute infection. So that's usually how it goes. So if you have, for example, any of these kind of, let's say, symptoms, then you're not gonna do an IgG antibody test. You're gonna do an IgM, because IgM is gonna really tell you, does this person still have an acute and ongoing infection? Because once you start having IgG antibodies, that is already telling you that, okay, you're gonna be protected, and you basically, it's not, most probably the virus is also gone by this point, because usually with an IgG antibody, you basically are able to remove it. And this is just to show you how good or how important antibodies are for hepatitis viruses, actually. Then we have hepatitis B. Hepatitis B is a very, I have to say, weird virus. It's basically it's a DNA virus, but it's basically its DNA genome is partially doubled. So you see here you have basically in kind of this kind of purple like color, the positive strand is all filled, and then you have the negative, which is blue. It's only up to here. So it's actually kind of broken, the DNA. It's not fully one circle. So it's in some sort of way partially double-stranded. So that's one. Two, it's basically enveloped, and basically it uses this hepatitis this hepatitis surface antigens, which creates your envelope. And also one other thing that is also weird from this virus is that it also does reverse transcriptase. It will produce, it's from its DNA, it's going to produce RNA, and then this RNA is going to be then brought back to DNA. So it has a polymerase that actually has a DNA polymerase or DNA-based polymerase action. So it's going to use DNA to polymerize, and then it's going to basically has also an RNA-based. So it's able to take this RNA and bring it back to DNA. So it's quite unique in that sense. So what basically happens is that, imagine this is your hepatitis B virus. It's going to enter, and once it enters, it's going to release. It's literally kind of partial double-stranded genome into the nucleus. The first step is to repair it, because now this is, as I said, it's kind of broken. So you repair it, then you create your RNA. RNA then basically is then taken. You basically start doing translation. But then once you have basically all your translation, all your proteins done, the RNA also serves as literally a template for DNA synthesis. So it literally is going to do this reverse transcriptase. So this actually explains why we can use these nucleoside analog that are targeting reverse transcriptase. Remember these ones that we use for HIV that are targeting the reverse transcriptase? Well, we can also use it also for hepatitis B viruses as well. They're quite effective. So as I said, basically the whole cycle is that once it basically comes in, it's going to bind, and basically it's going to release its capsid, and then it's going to release basically its partial kind of stranded DNA. It's going to fix it, produce mRNAs, and from basically one of the RNAs that it produces, it's going to be used to create your DNA, its hepatitis B genome. And one thing that is very interesting usually is that its envelope, this actually contains a lot of these hepatitis surface antigens, these proteins that are very, very specific towards hepatitis B virus. And this is going to be very important because it really is going to tell us a lot about infection and generally how to kind of, let's say, diagnose an infection. So transmission of basically hepatitis B virus is blood, sexual transmission, and prenatal transmission, and is extremely successful. Technically, you just need a tiny, tiny amount of blood to transmit hepatitis B virus. So in generally, its prevalence worldwide is quite high. I think there's like 300 million cases, sorry, of hepatitis B virus that is actually chronic. And we see this quite a lot in Asia. So if you look at basically your world map and we look at the endemic, how endemic is it? Just look at basically Asia, specifically China. You have quite a high frequency when it comes to the hepatitis B virus. And as I said, around 300 million people are chronically infected. So hepatitis B can stay there for long periods of time. And the issue with hepatitis B virus is that if basically becomes chronic, it actually can develop into hepatocellular carcinoma, so liver cancer. So generally, we used to look really a lot about hepatitis B as an oncogenic virus. It's a virus that induces oncogenesis. And of course, when we created the vaccine against hepatitis B virus, it was the first ever vaccine that was able to actually prevent a human cancer. And of course, it was really spread specifically in Asia to stop this huge endemic of basically liver cancer. So as I said again, with hepatitis B viruses, they do not cause death themselves. They do not cause direct cell death. It's the immune system. It's the T cells that go in there and keep attacking and destroying your hepatocytes. And these are the ones that are causing all these symptoms. So the thing about basically the hepatitis B infection, majority of the cases, you do not get chronic infection. So if you're a healthy adult, only 5% of people that get infected with basically hepatitis B get chronic carriers. If you're basically a baby or basically elderly or in a high risk, in a sense, basically where you don't have an immune system properly, you have a 90% chance now. So the jump from 5 to 90, it's quite massive. But this is telling you how important our immune system is, specifically cytotoxic T cells that are very important in controlling it. So what do we mean? How do you distinguish between someone that is acute and someone that is chronic? It's very simple. If it's basically looking at these hepatitis B surface antigens. If these antigens are present in your blood for more than 6 months, you're chronic. That's what basically we mean. So that technically is telling you that having the presence of this means that the virus is being replicating. So for more than 6 months, usually it will keep going. And in this state, usually the DNA of the hepatitis B virus, it can basically stay in the hepatocyte as an episome. Episome basically means it's literally in your nucleus. It's not integrated in your nucleus, but it stays there. So that's what we mean by episome. It's just there in the nucleus. Or some can actually integrate in the host genome. So this is what also makes them also chronic, be put in a chronic state. And basically what can happen is that, of course, two things can happen here, is that your immune system keeps getting bombarded, it keeps bombarding your liver, and this causes cancer. A chronic inflammation can cause, so a chronic inflammation can really cause cancer. It can drive oncogenesis. But also, the hepatitis B virus has a protein called HBx. And HBx what it can technically do is that it can actually bind to P53. So we know P53 is a very important cell when it comes towards it's a protein that's very important to stop oncogenesis. Every time our cells become oncogenic, P53 is able to recognize this and stop it and make the cell die. But what basically the hepatitis B virus can do, specifically HBx, is stop it. What can also happen is that when the hepatitis B virus is actually inserted in our genome, it actually can mess up with a lot of different other natural proteins per se, which can definitely lead to cancer. So one thing, of course, that we see of other than your cytotoxic T cells being produced, we get a lot of different proteins produced. And we get, let's say, two main groups of proteins. Ones that we call anti-HBs, which are basically the ones against the surface antigen that is found on the outside, the one that creates the envelope. And then we have other ones that are against the core antigen, and this is called HPC. So we call these anti-HPC. The anti-HPC are as useless as literally a fifth, basically, leg of a chair. You don't need it. How can an antibody go and bind to something that's inside a virus? Antibodies will only bind to extracellular proteins. They will never bind to something inside. But it can tell us, it can indicate actually that we have an infection. So generally, the real protective antibodies in our system is usually the anti-HBs, so the surface antigens. So generally, when we actually have a patient in the clinic that has a hepatitis B virus, we look at the blood markers. The blood markers can tell us a lot. It can tell us if the person is acute, if the person basically has a chronic infection, and basically, or has this person been vaccinated, or has this basically person has actually been recovering from the hepatitis B virus. So what we do is we basically look at four main types of things in the blood. We look at the surface antigen, basically the HBs, the surface antibody. So basically, do we have any antibodies against the surface antigen? The core antibodies, so these antibodies on the inside, and this E antigen. This E antigen is usually also found inside of your hepatitis B viruses, but usually these are indicative of something else. They're indicative of a very strong infection, which I'll later talk about. But we look at these four different types. So as we said, the hepatitis surface antigen is the first ever detectable viral marker, and it tells you the presence of an active infection. So if you find it in your blood, it tells you that the virus is there and is basically is replicating. And of course, it can be found in the acute, but also in the chronic infection, because if it's there for more than 6 months, this already tells you that you have a chronic infection occurring. And then you look at basically the antibodies. Do you have any of the surface antibodies? Because if you do, then you basically are in some sort of way immune to hepatitis B. So you're able to actually fight it off. So, and basically, as we said, 5% of people that that get chronic hepatitis B virus, they do not have anti-surface antibodies. These surface antibodies are the ones that give or confer protection. So it's extremely important. So we can either get them through infection or vaccination. So now there is the basically hepatitis B virus vaccination, which you can induce it. There's two basic, and then you have basically these core antibodies. The core antibody does not provide protective immunity. So as I said, it's literally as useless as a fifth leg of a chair. You don't need it. It's only useful for diagnosis. So you have two forms, the IgM and IgG. IgM tells you, is this an acute infection? IgG tells you if it's past or chronic infection. And one thing that we know from basically if you have the core antibodies, this tells you it's a natural infection. Meaning, usually when we give a vaccine, we do not vaccinate against the core antibody because it's useless. So we literally go just for the surface antigen. And then we have as the last one, which is the E antigen. The E antigen, usually we look at it because it tells you that there's a very high active replication and high infectivity. So if we see someone having basically this E antigen, this specifically the E antigen, it tells us this person actually has a lot of virus in the system and is highly contagious. So it tells us a lot about how will this person be basically interacting with other people. And then generally, if we have basically the antibodies against the basically the kind of E antigen, it tells us that we actually have a very reduced viral replication and lower infectivity. So how does basically the serology of an acute hepatitis B viral infection look like? Well, what you would basically see is that, first of all, as we know, the first thing that comes up are your surface antigens. This is positive. We have basically your IgM anti-HBC. So this is basically the core. It has to be of the IgM. And then basically, sometimes most of the cases, your E antigen also is often basically positive. But the key, the most important basically marker when it comes to seeing if it's acute, it's basically having a look that there's an IgM basically anti core protein. So it's not IgG, it's IgM. Then we have this window period between before getting an acute response and having then basically later on a chronic or basically. So what happens in this window is, as we see over here, these basically here you have basically your antigens. This is basically what this this window period, it refers that your surface antigens are basically disappeared. But what happens is that your surface antigens, antibodies, have not appeared. So there's nothing that's basically there. The only thing that's detectable are the IgM ones, but the IgG have not appeared. Only when they appear is basically when we do not have any more of this, basically, like it's quite almost zero. So this is called the window period, and where the only thing that you see in the blood, basically, is only IgM anti-HBCs, so basically the core, but you do not have any surface antigens and you do not have any anti-surface antigens, just you only see your basically your IgM anti-core markers. This is telling us that, okay, this person is actually going to make a recovery because there's no more surface antigens. You have antibodies, and it's just taking a bit of time to get these protective anti basically surface antigens, specifically of the IgG. So we call this the window period. And then if you basically have developed a chronic infection, what we see is that your basically surface antigens are positive for more than 6 months. We start to see that the core, basically antibodies, are of the IgG isotype. So these are the most important ones. And more importantly, you do not have any surface antigen antibodies. These are absent, because if you actually had them, then you would have had protection. But no, because you're chronic, basically the virus is still replicating, you do not have that antibody. It's absent. And of course, this causes chronic infection, which then leads to cirrhosis and cancer. So when it comes to prevention and treatments of the hepatitis B virus, prevention is extremely done through vaccination. It's very important. Technically, for example, me and when I was in my master's, when I was basically playing with blood samples, I basically was highly recommended, almost kind of forced by my basically by the hospital where I was working, to take the hepatitis B virus. Usually it's three vaccines separated by a month each, to make sure that you have a high titer of anti-surface antigens against basically the hepatitis B virus. But if you, let's say, contract chronic hepatitis B virus, usually you use basically this antiviral drugs that inhibit the reverse transcriptase, tenofovir and entecavir. These, especially tenofovir, is used for HIV as well. So this really helps to reduce the viral replication. Sometimes let's imagine that you are, especially a doctor, a nurse, or someone working, let's say, in the medical field, or you basically have slept with someone that has hepatitis B, a chronic infection. You do this post-exposure prophylaxis, and basically it means that you know that you have been at risk. So what you do is you literally go to the hospital and you basically give an infusion of literally immunoglobulins against hepatitis B, so antibodies against hepatitis B, specifically towards the surface antigen, and also you vaccinate yourself like quite directly right after your exposure. So now we have the hepatitis C virus. So the hepatitis C virus is an enveloped, single-stranded positive sense RNA virus. The thing about hepatitis B virus, of course, is that it does not use reverse transcription. It has its own RNA-dependent RNA polymerase. And it's quite a weird virus because it's single-stranded, positive sense. Why would it need its RNA polymerase? I still do not really know the real reason for that. But because it has its own RNA-dependent RNA polymerase, because of that, it's able to actually keep producing quite a lot of high genetic variability because RNA polymerases do not have this proofreading. They do not proofread. So what happens is that, let's imagine you're infected with hepatitis C virus. Literally, this goes into one cell. Suddenly, you create different versions of the hepatitis C viruses that have different mutations. We call this quasi-species, where you have multiple viral variants. So multiple hepatitis C viruses that have different mutations. So this is why you have quite a huge immune evasion and persistence of the hepatitis C virus. So the transmission of the basically hepatitis C virus, it happens again, literally similar to hepatitis B. It's usually through blood exposure. This could be intravenous drug use, blood transfusions, needle stick injuries. It can actually happen through prenatal transmission, through sexual and prenatal transmission, but it's less efficient. Usually, basically hepatitis C virus is done through blood exposure. And the issue with hepatitis C virus is usually that it's quite asymptomatic. You do not have quite a lot of symptoms. It's only when you start getting the very severe symptoms like cirrhosis or cancer that you start realizing that, I might be infected with hepatitis C. So again, just like I said before, basically hepatitis C viruses or hepatitis viruses in general, they do not do any direct killing. They just lead to basically a lot of immune responses, specifically your cytotoxic T cells. So the thing about our immune responses is quite ineffective because we have the quasi-species. We have a lot of these kind of multiple hepatitis C viruses that are just literally having mutations everywhere. So our immune system cannot keep up with the quasi-species. And unlike hepatitis B, where basically it's quite rare that you get a chronic infection, with hepatitis C virus, it's quite the opposite. The quite majority of the time, you're going to get it. Between 75 and 85% of the chance, you're going to develop a chronic infection. And this is mostly because what happens is that the acute infection is quite mild or asymptomatic. So you do not even realize. You don't think to yourself like, oh, I might have this virus and let me go get myself checked. No, you basically leave it untreated and it's going to keep going and going until it actually starts to do damage. Because the hepatitis B virus, as we see, we have antibodies, specifically the IgG anti-surface antigens that protect us. In the case of hepatitis C, we do not have a proper immune system. We have these quasi-species that are basically really limiting our immune responses. So what happens with basically hepatitis C is that we have literally the bombardment of our immune system towards our liver, and this can cause cirrhosis, so the scarring and basically kind of this kind of literally the liver looks like kind of this bubble wrap. It's very disgusting, kind of actually the look of it, which then can lead towards cancer. So cancer is one of the biggest things that you can get from hepatitis C virus. So as I said, one of the good things that have the hepatitis C virus is that it keeps changing its surface antigens. It keeps changing, has a high mutation rate. So antibodies and T cells are not able to actually keep up. But also the hepatitis C virus has its own little tricks. It can actually reduce your basically kind of interference signaling pathways. So you kind of you're stopping basically your innate immune system to recognize it. So hepatitis C viruses, usually the acute, usually it's asymptomatic, but if you do have, let's say, symptoms that are very mild, it's a bit of fatigue, nausea, and jaundice that you usually have. But then later on, it's literally silent for many years until a lot of the damage is already caused. And of course, the biggest ones is liver failure. Signs of liver failure start to pop up. And this can be quite a lot of dark urine, fatigueness, jaundice. These are the ones that usually start to pop up, and specifically chronic or persistent jaundice, because basically your liver is just damaged and a lot of bilirubin is literally going to your system. The diagnosis of the hepatitis C, usually what happens is you look at antibodies. First thing is you look at, do you have any antibodies against your hepatitis C? If you do, this is not still indicative that you have the basically infection. You go and test with basically a PCR with the hepatitis C RNA, because you could actually have the antibodies and you could be one of the lucky ones that actually has removed the virus. So these could be just as protective or just because they were there. So first of all, you first look at the hepatitis C virus antibody, then only if it's positive, then you basically do a PCR. Treatment, as bad as basically hepatitis C virus might sound, the good thing is that nowadays we have something called direct acting antivirals, so the DAs. And because of the DAs, we are able to cure 95% of the hepatitis C virus. So we managed to actually fight off hepatitis C. So this is actually very good because, as we said, these guys are chronic, majority of the times will become chronic and can cause cancer. So thankfully, our antivirals, actually, it's the most successful antivirals we've ever created for a virus. Question in the chat. What is someone improving risk? That's a very, very good actually question. Medium, yes, you do get actually false negatives. And generally, basically the doctor has to kind of realize that per se. Because imagine you have because as we said also in my primary immunodeficiency lecture, antibody basically deficiencies are only seen in your adulthood. So specifically kids or teenagers that get infected with hepatitis C virus, they can actually have a hidden immunoglobulin deficiency. So it can definitely give you a false and negative. Sorry, basically, yes, a false negative. So this is where you need to also have a look. Does this kid have antibodies? And usually, in general, you will be able to see if there's you should do some positive kind of what do you call it kind of tests just to see did you successfully remove serum, and within the serum did you successfully actually manage to have antibodies in there. So you do some positive basically kind of tests to see that is there antibodies. So in generally, if you skip that and if the person has some sort of deficiency, then yeah, you're gonna basically do the wrong diagnosis. Then we have the hepatitis D virus, which I never thought actually existed. This course really helped me a lot to understand it. So hepatitis D virus, why I never really heard about it, is because it's quite, in some sort of way, not really seen a lot. People are not really interested in it because it's a defective virus. It's a defective RNA virus that is also has an envelope, and its envelope is made from the surface antigen coming from the hepatitis B. So the hepatitis D virus, is not able to produce its envelope. It needs the co-infection, or it needs basically just hepatitis B virus there. So you do not see people infected with just hepatitis B virus. You see it only with people that are already infected with hepatitis B. So that's why it's quite a weird, let's say, virus. So this can happen in two ways. Co-infection, meaning that you're simultaneously infected with both hepatitis B and hepatitis D viruses, or super infection. So you are already chronically infected with hepatitis B, and then comes HDV later on. Super infection is more severe than co-infection. And superinfection basically can increase the risk of chronic hepatitis, cirrhosis, and liver failure because you're just having much more of your immune system being basically literally turned on. So, and generally, it's very important to see if you have hepatitis B chronically, you need to test, do you have hepatitis D virus as well? So, and generally, as I said, hepatitis D virus just increases liver disease and hepatitis B. So, and generally, it's very important when you see a chronic patient to always look at the hepatitis D virus. And generally, it just makes the rapid prediction to cirrhosis. And this could lead basically technically towards carcinomas. The diagnosis is quite simple. You look at basically the detection of the RNA or antibodies. In general, what you see, the first thing you do is you look at the hepatitis B virus. You see, do we have hepatitis B virus, specifically the antigens? Yes. Then you do basically your specific antibodies or RNA towards the hepatitis D virus. Because remember, the surface antigen is something that you see both in HBV and HDV. So it does basically, if you find this, you need to look at specific antibodies or RNA for HDV. The good thing about HDV is that we usually do not treat it. We treat hepatitis B because if you're able to remove hepatitis B, the hepatitis D virus is not able to replicate anymore. So usually HPV vaccination is the way to go with this. The final one, the hepatitis E virus, very similar towards your basically hepatitis A. In general, your hepatitis E virus is unenveloped, single-strand RNA. Sometimes we see it with some sort of quasi envelope, which means it's an exosomal membrane. Sometimes this is what we call quasi enveloped. It's different, but in generally, majority of the hepatitis E viruses are naked. So the naked hepatitis E viruses very, very similar to hepatitis A because again, they are non-enveloped. So they generally are much more resilient to basically your environment, specifically towards pH. And hepatitis E viruses are literally, you mostly see in waterborne epidemics. So basically where the water is contaminated. This is where we actually get majority of hepatitis E virus. Usually you can get it through also through food, but that's mostly hepatitis A. Hepatitis E is much more waterborne disease. So it's mostly waterborne. The biggest difference between hepatitis E virus and hepatitis A is it's kind of basically, what do you call it? Much more the specific risk group. So the clinical features are very similar to hepatitis A. You have your basically jaundice, fatigueness, elevated liver enzymes. But again, they spontaneously are quite easily removed. And you have a very good immune system against it. But the biggest distinguish between hepatitis E and A is the high mortality rate in pregnant women. Hepatitis A-E is very deadly in pregnant women. Hepatitis A is not, but the E is. So this is the biggest difference between the two. So generally, like other hepatitis viruses, the hepatitis E virus does not destroy your hepatocytes. It just causes literally bombardment of your T cells. So that's what typically happens. Usually, chronic infection is very rare. Usually, you have a very good immune system that's able to remove it, but it actually happens with immunocompromised individuals. So guys, this is the end of the hepatitis B, or just generally hepatitis viruses. Do you guys have any questions or anything that you guys want to go over?
Doctor, I have a question.
Tell me.
Why are antibody and immunological assay tests used in hepatitis, not like PCRs?
So that's a very good question. It's usually because because technically when it comes to a virus, you need to go to the source of it in general. So sometimes you can get it from the blood if you have high infection rates. You know what I mean? So technically, in general, if you want to know that if there's basically DNA or RNA in your blood, it means that there's a lot of infection. In some cases, it's not that maybe you have the infection, but it's not that high. It's not producing quite a lot. So you need to go to the source. So you need to go to the liver. So per se, to go to the liver in general is not the nicest thing. You don't want to be subjecting a patient towards literally basically a liver kind of what do you call it biopsy. So just take the blood. So then that's why it's usually better in that kind of sense.
Okay, thank you.
No worries. Any other question, guys? If not, guys, have a great weekend. I'll see you guys on Tuesday for the last class. Have a nice one.
Thank you,
Thank you.
doctor.
Thank you.
Thank you, doctor.
Thank you.